Hello guys, in this lecture I am going to cover a topic from PTSM subject from a unit second, that is a general principle of preclinical screening. So regarding experimental pharmacology and regarding the preclinical screening, there are a number of questions are asked by a number of healthcare professionals who are not from experimental pharmacological background. So we will justify those questions one by one, then we will move forward for the objective of our this presentation. Okay, so there are a number of questions. First question is, what is pharmacological screening? So in general, it is the process in which any new organic chemical are to be subjected to series of number of assessment for the safety and efficacy determination purpose. It is a process in which any chemical entity is subjected to the number of number of assessment for the safety and efficacy determination purpose. Okay. Another question is, what does a preclinical means? So pre means before. So before the clinical studies, that is before the clinical studies, that is clinical stand for human studies. Before the clinical studies, evolution of safety and efficacy of a drug into the corresponding suitable animal model or experimental animals. Another question is, what is the difference between preclinical and clinical trial? So it is very simple. Before doing a clinical trial, investigators conduct preclinical research using human cell culture or animal models. If the preclinical research is promising, they move forward with the clinical trial to see how well it works in human. Clinical trial happen in several phases, during which different questions are asked for the safety and efficacy purpose. Then what is the purpose of a preclinical testing? So the main goal of preclinical studies are to determine a starting or a safe dose for first in human study and assess potential toxicity of the product. That particular toxicity, it might be an adverse drug reaction, it might be any side effect, it might be any idiosyncratic effect. So which typically include a new in which the subject which are going to assess those include new medical devices, prescription drugs, and some diagnostic tools also. Okay. Another question is, how long does preclinical investigation usually takes? So preclinical research may take anywhere from 1 to 6 years. Researchers only take the most promising potential treatment through the journey to market. New treatment then go through several clinical trial phases. These phases test the treatment for the safety and effectiveness. This is all about these questions. So we will move forward toward the major objectives, major agenda of this presentation. That is to know the general principles of the preclinical investigation and preclinical screening. So let's get start with it. Objectives. So first objective of this presentation is to know what is preclinical study, to briefly overview a step involved in preclinical trials, to know that what are the type of screening, to see the route map of preclinical studies, and to learn in brief that what are the method of screening. So let's get start with it one by one. So we all are generally aware about drug development process. We have gone through this topic in M.Pharm degree syllabus. So I'll briefly recall this process. Here you can see the image one by one that this is a drug research. From here we will go for the estimation or determination of the number of chemical entities. here we will study the actual preclinical trials we will go through. Then after this we will move forward to the clinical trials. Okay, and after filing the NDA to the FDA, we will market this drug. Okay, that particular industry will sell this drug in market, and after this there is a starting of post-marketing surveillance. So here we need to file, we need to approve animal ethical committee approval. After preclinical study, here we need to file IND, that is investigational new drug application. After clinical trial we need to file a NDA, that is new drug application, and after this CDSCO or Drug General Control of India will give the permission to market this drug by overweighing that particular material which are these material which are provided by the overall drug research preclinical and clinical trial study. So we will overview this study. So in first step, that is the drug research, from various drug sources we get number of chemical entities, and from those chemical entity researcher try to find some few of the chemical moiety which are have promising structure, which might be having wanted or desirable pharmacological activity. So then these finalized chemical entity are tested for the safety and efficacy. So the humanitarian and for the protective decision we will screen these chemical moiety into the experimental animal which are having somewhere similar physiology as human, like for example SD rat, guinea pigs, rabbits, cats, etc. etc. To proceed preclinical study on animal, there is the law, namely Prevention of Animal Cruelty Act, which is established on 26 December 1960. Under this act, Animal Ethical Committee is there. We all are aware about Animal Ethical Committee. Okay, so this authority committee having their guideline for the animal studies nowadays, which we call it CPSC guideline for animal husbandry. CPSC stand for Committee for Purpose of Control and Supervision of Experiments on Animals. Actually it is the statutory committee which is established under chapter 4 section 15(1) of the Prevention of Cruelty to Animal Act 1960. So for the preclinical study we need the approval of protocol which we made. Then after the sanctioning of a protocol we proceed animal study on few of the compounds. Then after this we get some 3 or 4 compounds which are efficacious and safe also. So after this we can't able to predict the efficacy and safety on the basis of only animal study. So the solution is we need to go for clinical trials. So according to regulation criteria we should have to fill the application to the FDA, that is National Medical Authority. In India, National Medical Authority, which is nominated by WHO, is CDSCO, where the major person is DCGI, that is Drug Director that is Drug Controller General of India. Currently the DCGI of India is Dr. VG Somani. They will give us a permission for the clinical trial. After clinical trial we again need to file a NDA, that is new drug application, to same authority, and then our drug can enter into the market. And after this the post marketing surveillance will get start, that is the role of pharmacovigilance to report the ADR and some of the side effects or any another effects, monitoring of ADR and prevention of it also. So this is all about the drug development process. So moral of the discussion is, without preclinical study you can't able to get any further approval. It might be any IND, it might be NDA, it might be any marketing permission. Okay, so this is the importance of preclinical study. So from here we will start from our first objective. What is preclinical trial? Preclinical trial, a laboratory test of a new drug or a series of chemicals, usually done on animal subjects to see if hope for treatment really work and if it is safe to test on human. That particular definition is told itself that this is a laboratory test series of a new drug, that is number of chemicals, usually done on animal subject, that is animal study, to see if that hope for treatment really work and if it is safe, that is to check the efficacy and safety. Okay, this is all about preclinical trials. So steps in up to the filling of IND in case of preclinical trials are: first we will find a drug target. Okay, first we will find drug target, lead compounds, number of lead compounds. Then we will go for the development of a bioassay. It might be in vitro, it might be in vivo, it might be any invasive bioassay, it might be any non-invasive bioassay, it might be ex vivo also, in silico also. Okay, according to the availability. After this we will plan for screen the drug in the assay to check the purity of it and number of to check the safety of it and to check the efficacy of it. After this we will go for the establishment of effective and toxic doses by using number of bioassay like ND50, ED50, and we will check the actually safety of that particular drug. That is, will go to determine the maximal therapeutic dose and minimal therapeutic dose of it. We are going to calculate the therapeutic index of it. After this, if that particular procedure provide you the actual data, registered data regarding safety and efficacy of drug, you will be able to file for approval as an investigational new drug for the further process of drug development. That is, next step is clinical trials. Okay, to get into the clinical trials we need to file a IND and get a permission from national regulatory authorities, that is FDA in India. CDSCO will regulate it. This is the office of CDSCO in New Delhi. This is FDA Bhavan. Okay, so WHO already declares that CDSCO as a functional national regulatory authority. So again, next objective, screening of drugs. So screening of drugs itself suggests that a thorough investigation to, what is actually screening of drug is a thorough investigation to get pharmacological activity of a new or chemical undefined substances, investigate the function of endogenous mediators, and measure or define the toxicity and or unwanted actions. So here first point is get pharmacological activity of a new or chemical undefined substance. We will, by using a pharmacological screening, we will get to know that what actually pharmacological activity that particular chemical entity contain. Then investigate the function of endogenous mediators. We will able to find the function of endogenous mediators also by using pharmacological screening. then measure or define the toxicity and unwanted actions. We will able to find the adverse effect or the toxic doses of that particular drug. Okay, so this is the complete importance and the advantages of pharmacological screening of drug. Next is type of screening. This is again next objective of this session. So there are three types of screening. First is simple screening, blind screening, and program screening. Okay, so simple screening, as it suggests that so in case of simple screening, a researcher only try to find that particular substance activity. It is generally inexpensive and less time consuming, but it is not sufficiently accuracy in the result. There is no accuracy in result, and also there is this simple screening is done to find out the substances are active in single way. Okay, and for example, the hypoglycemic testing, that is ability of any compound to decrease or diminish the blood glucose level. This is the example of simple screening. Okay, activity in one way only. Then blind screening, is there any biological activity? Blind screening used for to check the is there any biological activity. So in case of blind screening, the researcher will try to find one chemical entity which kind of pharmacological activity. Okay, it is only for the series of new chemical substances with no prior pharmacological history. It is for complete new drug. That particular drug don't have any reported activity, so blind screening is for that particular drug. Here the new chemical entity or isolated natural product are screened. It demonstrates whether the new group of a substance is worthy for further attention. If that particular drug is not having any biological activity, not having any drug receptor interaction, any protein interaction in our body, so it is not useful for anything. So furtherly we will not entertain that particular drug, will not proceed further drug development process. Then another point is point out the most potent chemicals with interesting pharmacological activity. By using blind screening, we can point out the most potent chemical with interesting pharmacological activity. In blind screening, it requires planning and skillful execution of the test. Okay, it requires nature, economical, time, money, it requires all. Okay, next is the program. It is very best type of a screening. In the program screening, researchers try to find how or by which mechanism that particular active drug producing their effect, and also take a consideration about ADR or side effect report, side effect or safety of that particular drug. So it provides information what compound are active in what way. For example, system or organ specific study, a series of testing program is required to provide information on a compound on specific target. It is more limited than blind and a simple type of screening. It is very precise, and for doing these procedures, precision is also important. Precision expected because it is a program. So these are the screening method and carried out one after one respectively, as arrow indicate in descending manner. Here you can see. Okay. So we will move forward towards the route map of a preclinical trial. It seems like when we talk about it, it seems like a little bit complicated. Actually it is not complicated, it is really simple. So Actually, it is really simple. In preclinical trials, there is two methods according to the natural nature of the study. That particular researcher or the investigator will select one of them. Like first is in vitro and second is in vivo. So when we talk about in vitro study, it is very simple study outside the in vitro study means outside the body or physiological system. For example, in vitro bioassay, which we have already performed in degree level to find out potency and activity of number of drugs like acetylcholine, histamine, atropine. We have already seen the antagonism, we have already seen the potency, how to calculate the potency, how to calculate the concentration, okay, by using that various type of bioassay like interpolation bioassay, bracketing bioassay, 4-point, 3-point, graded type bioassay, endpoint bioassay. Okay, so and another point is in vivo, that is vice versa, inside the body. So by administering any drug moiety by various route, it might be oral, IP, IV, subcutaneous, drug will get administered and it get absorbed, then it get into the blood circulation, and then we will access the activity of that drug by using number of model of experimental animal model, and we will move forward for the number of parameters, like for example analgesic to study the analgesic activity. We will move towards the analgesic activity, like to check the thermal hyperalgesia. We will consider the one test like analgesiometer, hot plate, then Randall Salito. There are lots of method to screen the analgesic activity. Actually depend on what we are going to check, either pharmacological activity or the safety. Okay, after the finalization of the safety, dose or range of it, we can call it as a therapeutic index. Here in case of in vitro, there are a number of example I have put in, that receptor binding assay, enzyme inhibition, enzyme induction, cytotoxic activity we can predict by in vitro assay outside the body. And here in vivo, there is we will check, might be pharmacological study or might be toxicological study, like safety. After the finalization of safety, dose or range of it, we can call it as a therapeutic index by calculating LD50 or ED50. In case of toxicology study, we can calculate the LD50 and ED50 by using in vivo biostudy, and furtherly we will go for the estimation of therapeutic index. Generally, less therapeutic index, the drug will be potent and toxic. There is a large range of therapeutic index, drug is very safe. Okay, get into in your mind. It is very simple concept, therapeutic index. So we will proceed for estimation of minimal and maximal therapeutic dose. For this purpose, we will divide the animal group into the test 1, test 2, test 3, etc. by like test 1 having low dose, test 2 having group medium dose, test 3 having group higher dose, etc. and compare our all the test dose activity output with universally accepted standard drug. As we studied in degree, the principle of bioassay is all about comparison of standard with test, and according to this output we can get our better therapeutic dose. Okay, so in case of toxicology study, we will go for the safety estimation, safety assessment, and in case of pharmacological study, we will go for the efficacy estimation, and furtherly we will move forward to check the dose conversion according to the safety and efficacy. So we will select that particular dose which is safe also and produces therapeutic activity. at maximal therapeutic dose. Okay, and after this it will leads to determination of starting dose for the clinical trials. So according to this output we can get our better therapeutic dose which is efficacious and don't having any toxic dose. This is how actually preclinical study is carried out. This is the route map of preclinical study. So there are a number of terms in the preclinical studies. So type of studies, generally it is called as type of studies, preclinical type of studies. So what is ex vivo? Okay, so ex vivo literally means that which takes place outside an organism. In pharmacological language, the ex vivo refer to experimentation or the measurements done in or on tissue from an organism in external environment with animal alteration of natural condition, like for example bioanalytical studies, for example any cell line studies. Okay, this is the example of like cancer study for cell line. If any person is doing a cancer study, he can work on cell line, okay, by growing some media. Then another is in vitro. You all are aware about in vitro. Actually we all done this procedure, that is procedure of student organ bath mounting of tissue, okay, and this biopsy procedure and at the degree level. So in vitro study are performed with the some of the microorganism, cell or biological molecules, that is biological tissue, outside their normal biological context, colloquially called as test tube experiment. These studies in biology or pharmacology. Okay, so here in vitro means outside the body that particular tissue is mounted, and we will test this particular activity on this particular assembly setup. Another is invasive. So what is invasive? The procedure that invades or enter the body, usually by cutting or puncturing the skin or by inserting instrument into the body for diagnostic or estimation purpose. So if you need there is requirement of any incision for any parameter or any estimation, pharmacological estimation, for example carotid artery cannulation, if you want to record the ECG, if you want to record the pulse rate, if you want to record the beat per minute of heart, you will cannulate this carotid artery, which is a divisions, both left and right divisions from aorta. For this you have to make an incision at this place, okay, and you will, by clamping, you will easily able to cannulate, and by this particular pressure, by using sensor, you can detect on your screen by using power lab. So this is the procedure of carotid cannulation. First you have to remove the hair, and then incision, and try to find out the carotid artery which is actually been at the esophagus. It's depend on you want to you want to cannulate left artery or right artery. After this, here just uplift by using spatula this artery and try to cannulate by this cannula. Here you can see by this cannula, and just tied with the biodegradable threads. And after this, by using catheter, that ultimately sensor will sense the pressure of that particular artery blood pressure. Non-invasive, look, in carotid cannulation or femoral cannulation, we need to make an incision. For that we assess ECG pulse rate. So without incision we can easily assess ECG. Okay, so here by using two electrodes, okay, by using electrodes we can easily place those electrodes on particular left and right paw, and ultimately we will able to get the ECG by anesthetizing that particular SD rat. Okay, and we can get ECG by using non-invasive method. So what is meant by non-invasive? Non-invasive procedure do not involve the tools that break the skin or physically enter the body. Okay, then in silico method. What is in silico method? In silico experiment is one performed on computer simulation. The phrase is pseudo-Latin in silico, that is in silico referring to silicon in computer chips. So it's all about computer aided animal tracking devices. Some of the cancer study you can easily predict by computerized method that particular drug is having which kind of effect on which organ. We have already covered in vitro, what is in vitro, what is in vivo, what is in silico, what is invasive, what is non-invasive. So these all are the methods for preclinical studies. Okay, so thank you. I hope you find this presentation useful.